Start recording. All right, we are good
Yeah.
to go.
Well, thank you everyone for having me here today and coming online to sort of listen to me. Let me quickly introduce myself. So I'm Raghav. I'm a PhD candidate, as Rana told you. I also do a few things on the side. I work as the director of bioinformatics at this thing called Healthy Longevity Clinic, which is this clinic that kind of helps individuals in sort of living healthier, longer lifespans, essentially, by providing more cutting edge interventions. And I also work with this thing called Longevity Tech Fund, which specifically invests in therapeutics and diagnostics that can help people live longer, healthier lives. So again, the whole premise is of all of this is that till now we have been our whole sort of medicine system has been very reactive in nature. So what happens is we typically get diagnosed with the disease and then we get treated for it, which is great, right? We have this paradigm has really helped us. So we get diagnosed with cancer and then we get sort of therapies for it. We have CAR T therapies, and we have sort of really allowed us to really extend lifespans in a lot of ways. But can there be a paradigm of medicine where we just totally prevent the occurrence of cancer or the occurrence of diabetes or cardiovascular disease in the first place? And that's what we call as preventative medicine. Um, and to do that, we have to dramatically change how we practice medicine and how we think about medicine. We need to have new biomarkers that can allow us to not track uh, diseases, but track aging itself. Um, and we need to have interventions or therapeutics that don't target specific diseases, but rather target the underlying physiology of aging itself and potentially is able to slow it down, stop it, or even reverse it. And that is the whole premise of my research, that how do we build these biomarkers and therapeutic interventions that can allow us to quantify aging and potentially slow it down, stop it, and reverse it. Um, so that's what my research is on at Yale. But let me, before I go into like how and what I am doing, let me tell you how I got here in the first place, because I think that would be of a lot of interest to this kind of to this conversation, because a lot of you in some ways are trying to think about how to get into bioinformatics and computational biology. And I'll tell you this, I'm not a bioinformatician by canonical training prior to my PhD. So let me give you a slightly long 5 minute story, and then I'll go into my presentation. So I started off my PhD. I actually started off my sort of whole journey of academia in during my undergrad as an electronics and communication engineer. So I had no experience in biology or in sort of computational biology for that matter. But what I did have was a really strong interest in studying biology and biotechnology. And I can tell you that because during high school, we had this course. By the way, I did my schooling and my undergrad in India, Delhi. What we had was you could pursue like physics, chemistry, mathematics, biology, which is the equivalent of your AP courses here. Um, and then we had this very odd course called biotechnology, which very few and very rarely people took because no one found it very interesting. And I was the odd kid in my school, the one person in my school who actually took this course. So just because of me, they had to like offer this course because I was just so interested in it. Um, I was actually really interested in biotech during my high school days, and then when it came to pursuing it during college, I just did not pursue it because there were no options for it back home in India. So unfortunately, the way the Indian ecosystem is structured, it is very heavily focused on sort of computer science and electronics engineering. As you might know, Indians are known for their computer science skills and their electronics development. We have a flourishing computer science industry back home, and and which makes sense. And so, from a very practical standpoint, it made sense to for me to sort of join one of the money making this thing. And my parents were the ones who suggested that I do that, and I as a young 17-year-old impressionable 17-year-old, I made that choice. And so when I was in college, I really didn't want to pursue computer science because I wasn't really personally excited by it. Digital circuits and that kind of stuff. Circuits was more my cup of tea, so which is why I pursued electronics. Um, when you study electronics, actually, you learn very deep amounts of mathematics, like really like you're learning like a lot of differential equations, you're learning a lot of sort of heavy mathematics that is required to understand sort of signal processing and all of this stuff. Just FYI, those skills are very transferable. Those deep mathematical skills are very transferable to any field that you might go into, and which is exactly what happened. So, come my third year of college, this company came to um, for an internship, offering an internship in computational biology. It was a startup. Funny story. They had an interview process. In the first step of the process, they were to give a presentation, and everyone was to attend it. And I attended it. I asked a few questions, and then the second step was essentially that you had to give like a computer science test. I failed miserably at the computer science test because I didn't know computer science. But as it turns out, because I was answering the questions, they were really impressed by me. And they called me later on to just say, like, hey, you seemed really interested. And so we want to talk to you a little more. And I was like, okay, I guess that's fine. And they talked to me and they felt that I was really passionate about the field and they could teach me the computer science part because it's really difficult to, according to them, and that's what they said, it's very easy to teach someone computer science, but it's very difficult to teach someone to how to be passionate about something. And um, and they felt that I was really passionate about the field in general. And so I joined the startup for the summer. And the startup, we took part in this thing called this Dream Cancer Challenge, which is like this international challenge where you compete against teams from Yale and Harvard. And just FYI, we were now it sounds great sitting here at Yale, but back then I was like this really small unknown college back in Delhi, India, right? So competing against teams from Yale and Harvard felt very, how should I say, terrifying. Uh, but we came second in this competition, just based on sort of pure skills of learning data science and stuff like that. And I think that was a very transformational moment for me because I learned something that was, I think I can be good at this. Uh, Unfortunately, by the end of my summer, this startup failed. Uh, and and but good, luckily for me, I had a, I had this really good prize under my belt. I got offers, really good offers from sort of McKinsey and Goldman Sachs to join as data scientists as at the start of my fourth year, which is like the senior equivalent in college back home. And so I was like, well, I could start those jobs. Interestingly, towards the end of my fourth year, my founder from one of those companies decided to start a new company uh, called Elucidator. My boss, essentially, from that previous startup. And he was like, "Hey, do you want to come and join here?" And I was like, "I don't know. I just got these really nice offers, and they're gonna pay me a lot. I don't know if I want to leave these companies and like join you guys." But he was like, "Why don't you come and like try this out for a bit?" And I was like, "Well, I'm in my final year of college. I have these jobs. I'm pretty much done with my courses, so I can." Like spend 2 or 3 days just like spending doing time here at the startup, and the rest I can do my courses. So I used to do like 2 days at this startup and the remaining days at these courses. And turns out that went amazingly. And at the end of my fourth year, I decided to not join those big companies uh, as a data scientist, but instead join this startup, Elucidator, as one of their first employees. Um, that startup now is valuated at upwards of hundreds of millions of dollars. And uh, and what it does is essentially it agglomerates publicly available data and cleans it up. Publicly available biological data, it cleans it up and then sells it to pharmaceutical companies for doing drug discovery, target discovery, and a lot of machine learning insight generation, and makes a lot of money out of it. And I spent my like 4 years about there doing data, leading their data science division at one point, and then eventually leading their product arm when they started developing a product. But at one point, I finally decided that I was kind of hitting a ceiling at the startup. I canonically was not trained as a computational biologist. I did not have the training, I did not have the knowledge. Here I was, an undergraduate who was, for lack of a better term, talking out of my derriere uh, to these really smart scientists at these pharmaceutical companies about uh, all of this data, where I actually had no idea about what some of this data meant. Um, and so for me, what was critical then was I had to go back and study. Um, and so I decided that I needed to go back to college, and I needed to go back to academia and do a degree. And that's where I decided to come back to academia, and one and sort of Yale happened. Um, I was initially planning on doing a lot of cancer biology, but then interestingly, one of my uh, my mentor, my first mentor, Dr. Morgan Levine, was giving a talk here at Yale, and was um, she was talking about aging as a disease, as a treatable condition, and that really changed my perspective on, hey, if we can solve aging, we might be able to solve cancer by itself. Uh, and that really sort of transformed, hey, this is probably what I want to pursue. And I did that. I joined Morgan's lab, and it's been a transformative journey. Morgan actually now has moved from Yale to this really big startup called Altos, which is a $3 billion startup that is just trying to solve aging. Her postdoc actually became an assistant professor now, and whose lab I am now. His name is Albert Higgins Shen. Uh, oh, very cool, Raul. And my 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 first project called Systems Age, well, and I'll talk about this a little bit. It quantifies aging in 11 different organs of your body from a single DNA methylation blood draw. And that got patented and licensed out to a company, which honestly is very unreal to me, because coming into academia, I was very naive almost, thinking that academia can never be translational. Uh, but seeing that within my PhD lifetime, and I've just been here for what, 4 years, uh, within like 3.5 years from the conception of this project to like the paper being out on bioarchive, we got this licensed out and patented, and that's just too too big for me, honestly. Mostly, I've never seen that happen for a lot of PhD students, and I'm just too shocked that it happened to me. So I'm quite grateful for it. Um, and I'll talk a little bit about what my other research is, but in general, what I'm speaking here is that a lot of this happened by coincidence, but a lot of this was also purposeful. That I made some active choices. Thank you, Diana. A lot of that, this is also purposeful in the sense that I made some active choices on what I wanted to pursue and what I needed out of my academic and professional career. And with that, I'll now like to shift a little bit into my actual research and give you like guys a little bit of a preview of what I do. But before that, if there are any questions, I'd be happy to answer them. If not, I jump into my presentation. Um,
I have a quick
yes,
question. It's
yes,
more out of curiosity. Let me
yes.
turn on my camera. Um, Once you finish your PhD, do you plan on going back to the industry or like to work for a company, or do you plan to stay more in like academia, research?
That's an excellent question. I'm literally having conversations with Morgan and some of my other mentors about this. I think there are pros and cons to both of them. With academia, you have a lot of flexibility, but I'm afraid whether this ability of having the translational part, I think I was partly lucky and partly I had to push for it, right? Along with my mentors, of course, right? But I don't think that happens all the time. Uh, uh, so I think I will, if I stick around in academia, that is something that I will have to actively work upon. As opposed to industry, the ultimate goal is translation, right? So if so, then I'm not actively thinking about it constantly. That is what the goal is. Then everything is about translation. So I'm personally really excited about translation. Coming from a startup environment, I really enjoy translational work. So that is one thing, but academia also offers a lot of sort of flexibility on pursuing a lot of different research ideas, which I personally really enjoy. Uh, Honestly, that is something that had not considered when entering academia. When entering academia, I was very sort of clear cut in my idea that academia is going to be a means to an end, and the end being that I'm going to go back into industry.
Right.
But having spent 4.5 years now, I feel a little more having mixed feelings now that maybe I should stay back. So honestly, I don't know yet.
Yeah.
Um, it's quite contradictory. Most PhD students feel disillusioned from academia after spending like they come with like these big hopeful eyes of being like, oh, I'm gonna change the world in academia, and then they feel disillusioned and go back to industry. It's quite been the opposite for me. I came in with this idea like industry is the way for me, and now I'm like, I don't know. So I don't know honestly at this point. I think it for me it has to be a good mix of translational ability, but also a certain amount of freedom to do the kind of research that I want to do.
Thank you.
Yeah, thank you.
It's
Um,
such a hard decision to make.
yes, for sure. It's
Yeah,
definitely a hard one.
but I second Diana, and I also want to say congratulations on your patent and your license. This is
Thank
really
you.
big accomplishment. Yeah.
I appreciate it. Thank you so much. Yeah, as I said, it's been both perseverance as well as luck in some ways, a combination of both. Um, Opportunities like this can come and you just need to hold on to them. But um, yeah. Uh, but again, the fact that you have to be it's 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 weird because you also have to be like an opportunity like this is rare to come by, so you just have to stick by it as and when it comes. Uh, okay, so let me we have about 30 minutes more, so let me use that to sort of talk a little bit about my research actually and how we are really leveraging AI a little bit. And I'm not going to go really crazy on like the details of it, but more sort of give like a 10,000-foot view of like how we are using AI and how that is really being transformative in some ways. And when I say AI, some of it is going to be really sort of the nitty gritty next generation stuff, but some of this is also going to be using some really next generation bioinformatic pipelines and sort of cumulative data annotation. And I'll talk about some of that as well. So let me share my screen right here with you guys. Okay, can everyone see my screen here?
Yes.
Yep,
Mhm.
perfect.
Yes.
Okay. So, I kind of want to start with a very obvious statement, which some of you may know, some of you may not know, but Geroscience is making headlines. And so I want to actually go to this NPR article, which is that this drug called metformin, if you might have heard of it, it's a very common diabetes drug that is given to a lot of people. It's now being potentially run in a trial called the TAME trial, and which may be one of the first therapeutics. It's been claimed to potentially extend lifespan and health span. It decreases not just not just it not only does it decrease the first occurrence of diabetes, but also decreases cardiovascular disease events and as well as Alzheimer's, but could potentially generally extend lifespan as well. All of this is in retrospective studies, but people actually want to run a clinical trial to show this now. And there's this TAME trial that is being run or is being at least funded to run this. So clearly, there is sort of the more scientific part of this, like potentially trials being run and trials being set up to verify if already known therapies or geroprotectors, as we call them, might be extending lifespan. But there are some more interesting parts as well. So the likes of Kim Kardashian are now trying to measure their biological age. I don't know if you have seen this, but in season 4, episode 10, which is the finale, actually, they measure their biological age and try to see which of them is the youngest. And guess what? Khloe Kardashian is apparently the youngest, which I did not expect. Um, and then there are sort of these more sort of biohacker people, which again, if you're in the aging community and if you're on Twitter actively, you might have come across this person called Brian Johnson, who is kind of this multimillionaire CEO who spends like about $2 million to $1 million a year just to record every part of their body and takes a gazillion supplements to stay really young. Um, so in different ways, sort of we're seeing uh geroscience being in the headlines, whether it be reality stars or actual science, or whether it be sort of biohackers in different ways, essentially. But as you can see, the commonality across this is that everyone's trying to slow down aging. They require biomarkers and they require geroprotective interventions, right? Um, and so, as a field, as a scientific field, what we really need to do is think about this harder as a problem. And we cannot sit on our laps, like we cannot sit by ourselves thinking that we. I think the field people are moving forward. We need to move with them at this point. So we can't just like be there and sit still. We really need to move quickly on this. So, to the outside world, it almost seems like people think that this problem is solved. Um, like for us, as some of these people that are on this on the Zoom call, we might understand this problem is not solved. But if you go to an average Twitter user who follows someone like Brian Johnson or maybe Kim Kardashian, they might believe that this problem is solved. Because to them, it feels like someone like, and I'm using the term Brian Bonson as a joke here, goes to a longevity clinic. They get their clinician measures their biomarkers to quantify their aging. Based on that, they are suggested sort of some biological interventions. A few months later, they are assessed for their biomarkers again, and then sort of JB leaves the clinic feeling rejuvenated like an 18-year-old. But the thing is, at each of these steps, there are a lot of challenges. So, for example, at the step of measuring aging biomarkers, right now, what we have are a lot of biomarkers that are essentially telling us whether you're 5 years older or 3 years younger. They're not insightful. Not a clinician can't really do much with an aging biomarker if they just tell you that you're 5 years older. They need something more than that. They need to know something about your systems, your organs, so on and so forth. And then when it comes to interventions, we don't really know which interventions are really geroprotective. We're kind of playing, it's a little bit of wild, wild west here. We don't know which interventions are geroprotective. And more importantly, an intervention that might be geroprotective for you might not be geroprotective for me, right? So again, there is the potential for it being a little more specific. And then at the last of it is responsiveness. So of these biomarkers. So even if these biomarkers can predict future disease and future health outcomes, if they are not responsive to interventions themselves, that is, they do not decrease when an intervention is affected upon, then what is the use of actually having this biomarker? And we don't know if any of these aging biomarkers that we have are responsive or not. So a lot of challenges sort of remain in the field. So let me talk about how we're trying to solve some of these challenges, at least from the perspective of our research. So we have a lot of sort of these biomarkers that can come from molecular, physiological, digital. One of the ones that I work on a lot is sort of epigenomic, and within epigenomic, sort of DNA methylation-based biomarkers. They've been shown to be sort of very prognostic. So DNA methylation, as you might know, is on a very simplistic level is essentially sort of these methyl groups that have been added to sort of DNA, and they can essentially regulate how much a gene is expressed or not expressed. So if you have more methyl groups on the DNA, the gene is expressed less. If there are less methyl groups, the gene is expressed more on a very simplistic level. And there are some more complicated interactions as well, which I'm not going into, but on the very basic level, that's one of the ways DNA methylation works. So at this point, the biomarkers, as I said, are very unidimensional. They take your whole blood DNA methylation. So it's either actually just a tap of blood, you swab it, and they are actually these actual machine learning algorithms. They're very simple. You have about sort of a simple array of DNA methylation will give you about 1 million sites, close to 1 million different DNA methylation sites from your DNA. And they take that information and compound it into one unified score. And that unified score tells you, we call that unified score as a clock, right? That clock tells you whether you're 5 years older or 3 years younger, right? So what it's essentially telling you is, are you aging faster? Are you aging slower? On a very simplistic level. That's what this is telling you. But for a clinician, this is really cool, right? Now from a simple blood drop, right? You're able to tell not just not only you're telling you, well, chronologically is the number of birthdays, number of candles on your birthday, right? But now you're able to tell your biological age, right? You're able to tell how quickly you're aging or how slowly you're aging, right? Which means how quickly you may die, which sounds very morose, but that's the reality of it, or how quickly you may get sort of aging related diseases, right? um, but that's insightful, but not insightful enough for a clinician to actually act upon it. You need something more so that a clinician can prescribe you interventions, can suggest you what to really do about it. So what we did was we built something called systems age. Um, and what this does is something very trivial on a very simplistic level, that instead of measuring whole body age, right, it divides that whole body age into different systems, such as brain, heart, lungs, kidney, liver, sort of 11 different systems like this, right? So instead of getting one score for the whole body, now you're getting 11 different scores for each of these organs, right? And we combine these 11 scores eventually into a whole body score as well. The way we do it is, again, using just a drop of blood or just sort of whole blood DNA methylation blood draw. You can predict changes in all of these organ-specific blood biomarkers. And once you can predict the changes in these blood biomarkers, you actually don't need those blood biomarkers anymore. And you can build sort of these clocks. Now, I'm actually being very hand-wavy with the training pipeline because the training pipeline is actually very complex. It's a huge 6-step machine learning pipeline where you first do dimensionality reduction on one end for the DNA methylation, and then you do dimensionality reduction for each of the biomarkers. You predict the DNA methylation dimensionality reduced species with using the biomarker PCs. And then you combine each of them using a mortality predictor that gives you 11 scores. And those 11 scores are then again combined using another mortality predictor to give you the whole body score. Clearly, you can see this is a fairly complicated pipeline to build this. But on the more simplistic level, what this has essentially allowed us to do is tell us why are you 5 years older? So if your whole blood DNA methylation tells you you're 5 years older, what this allows us to do is tell you the reason you're 5 years older is probably because your heart is 11 years older than it's supposed to be. Now that is really insightful because now a clinician can actually potentially act upon this. They can potentially prescribe you interventions that might slow down heart aging. And that, in my opinion, sort of goes a lot further than just knowing that you're 5 years older. And so this is what we essentially sort of patented and sort of got licensed out. And this is available as Symphony Age to clinicians and patients via True Diagnostics, which is a company. But the question is, can we go deeper with just DNA methylation? So this is something new that I've been working on. It's called CPGPT. Again, as you can see, it's a fairly complicated framework. I'm not going to go into a lot of detail here. But on the basic premise of it is, it does something similar to the systems age framework, where it can predict mortality, but something new, which we call as epigenetic biomarker proxies. So what you might ask, what are these epigenetic biomarker proxies? Now, these epigenetic biomarker proxies are essentially, you can predict thousands of proteins and metabolites measured just from one methylation blood draw. So just from one DNA methylation blood draw, you can quantify hundreds of proteins and metabolites. Now, the reason you can do that is because these DNA methylations are really powerful in predicting sort of these individual proteins and metabolites. And that's what we have been able to see using sort of these CPGPT-based GPT-based methodologies, which is really exciting. So why is this really exciting from a translational perspective is because we can go a lot deeper now from just one whole blood DNA methylation blood draw. So we can tell the clinician now, or a patient for that matter, the reason you're from your blood draw, you're 5 years older. And the reason you're 5 years older, because your heart and lung is aging really high. The reason your heart and lung are aging really high, because you probably have high homocysteine, cholesterol, and troponin. If you had to actually get all of this done in practice currently, you would have had to get multiple different blood draws and multiple different tests, and potentially it would have been super expensive as well. But instead now you can get all of this information from one single blood draw thankfully using some really impressive machine learning algorithms. And that, in my opinion, is the real power of using artificial intelligence in this approach. So I want to jump into sort of the next section, and we're kind of running out of time here. So how do we define effective geroprotective interventions? So one of the challenges here is that we do not know which interventions are geroprotective, because we just, in the studies that are run, publicly available studies that are run, there is no standardization of which biomarkers are quantified. They keep on running different biomarkers, especially DNA methylation, epigenetic clocks. So what we did was we used sort of a lot of publicly available data. The way we annotated a lot of this publicly available data was through machine learning algorithms. We took about 50 plus interventional studies in humans, longitudinal longevity interventional studies. So these are clinical trial data and sort of interventional study data, and calculated 110 different epigenetic clocks or DNA methylation aging biomarkers. And so each little box you're seeing here is like a t-test of before and after the intervention for that biomarker. So essentially, each of these rows are essentially one clinical trial here, and each of these columns is one biomarker. So you're seeing sort of 50 different clinical trial data and sort of 100 different sort of biomarkers in one heat map. Now, this is all really exciting, and we can compress this data to really sort of look at how these interventions are really changing a bunch of epigenetic clocks. And what we start to see is we look at some of the most prevalent or well-known epigenetic clocks and sort of perform a one-way t-test on the effect sizes. And we start to see a really interesting trend that there are some interventions that significantly decrease epigenetic age. That means that they are potentially causing a slowing down of epigenetic aging and potentially biological aging by that extension. These include hyperbaric oxygen therapy, metformin that I just talked about, some anti-TNF therapies, vegan diet, and smoking cessation, low-fat diet, so on and so forth. While some very well-known interventions like senolytics might actually be increasing epigenetic age. And of course, there's more to be studied here. And I should repeat, all of this data is in humans. We also see some overarching trends around which of these we can categorize these interventions into lifestyle, pharmacological, supplements, and medical procedures. And we see generally pharmacological interventions seem to be driving the most effect. Lifestyle, the next, and then supplements and medical procedures don't seem to have much of a significant effect in decreasing epigenetic age, which in my opinion kind of tracks. Supplements are not that great. I'm going to skip over this part, but maybe go through it very quickly. We can look at very specific effects of interventions as well, because now we have the ability to look at sort of organ-specific ages So something like metformin has multiple different effects across the body, across multiple different, or something like hyperbaric oxygen therapy specifically targets the lung. And why this is really exciting is because now we have the potential to match these interventions using system specificity. So if someone's aging really high in their lung aging, for in this case, for example, here, you can then prescribe them hyperbaric oxygen therapy, which specifically targets lung aging, which is great, in my opinion, because previously you would have just prescribed them, hey, you should just go try metformin, which might have not affected them at all. Because you might have come up with like, oh, they are 5 years older than they are supposed to be. Let's just give them the generic aging therapeutic metformin, but it might have no effect on them. So this will essentially allow for personalization of therapeutics. And the last challenge, let's talk about responsiveness. So again, using this particular study, we can actually look at responsiveness as well. And we have so many biomarkers here, but I'm looking at only 15 of them. And from these 15 biomarkers, we can see that only a handful of them actually decrease across the board for these 50 interventions significantly, which to me is very striking, that maybe not all of these biomarkers, not all of these epigenetic clocks should be used in clinic. And at this point, as I said, it's the wild, wild west. People pick and choose which clocks they should use for their trials, which clocks they should use for their personal health. But there needs to be a standard set as to which of them should actually be used for sort of by doctors, by clinicians, for the purpose of clinical trials, so on and so forth. And hopefully this kind of information will drive that. I quickly want to talk about like how we're sort of using some of this information at the longevity clinic, because again, translational aspects are really critical to me personally. This is just stuff about the clinic itself, but these challenges are really exciting, and we're trying to address this at the clinic. We're using explainable biomarkers for patients, mapping targeted interventions to patients at the second challenge using explainable biomarkers, and then using reliable next generation biomarkers that show response to interventions. So I want to show an example of what we call as an N=1 trial. So you might have heard about the standard of what is RCT trials, randomized control trials, but there is another paradigm of trials which is called N=1 trials, where you literally run a trial on an individual, and go and read about this more. And it's a really interesting paradigm to really personalize medicine. And I would suggest it's a really exciting read, because as we move into personalized medicine, these are the kind of trials that would really allow us to sort of individualize these interventions using sort of these novel machine learning methodologies. So let me talk about a case study that we did here. So there's this individual with a chronological age of 51 years. They have a history of autoimmune disorders and such as arthritis, and BMI of 31, and the substance use disorder behavior that they are a current smoker. So as you can see, some of these behaviors would explain their accelerated aging. So we have sort of aging in their different organs as measured by systems age, the score that I had developed. And on this radial axis, you can see which systems are accelerated versus decelerated. So you can see the musculoskeletal system is accelerated, which potentially can be explained by arthritis, as well as immune aging. You also see sort of something like lung aging is accelerated, which can be explained by their smoking status, as well as metabolic aging is accelerated. They have +5 years, which can be explained by their obesity status. So this individual was prescribed smoking cessation, of course, which is very obvious, nothing novel about that. Intermittent fasting to decrease their metabolic and musculoskeletal aging, as we know from sort of our specific interventions data, and metformin, which has a multi-system effect. They were also prescribed NMN, which we do not have data for here, but it is also a therapeutic, which is supposedly has multi-system effect as well. After 1 year, They had strong medication adherence to sort of IMF, intermittent fasting, metformin, and NMN. They continued smoking cigars, but decreased their smoking cigarettes. They have lost about 9.5 kgs and had qualitatively less arthritic pain. But let's actually look at how their sort of biomarkers changed, right? Because they significantly decreased on their musculoskeletal aging. Their metabolic aging went down, as well as their immune aging went down significantly, which is great. That is what makes sense given the interventions we tried. But we also see decrease in their multiple different systems as well, based on the fact that we gave them a therapeutic like metformin, which does have a multi-system effect. Which is also one interesting thing we see is that lung aging doesn't go down, given the fact that they do continue smoking on cigars, even though they quit smoking cigarettes per se, which kind of makes sense. But as I said, what's most important is sort of their qualitative experience. Qualitatively, they have less arthritic pain in their last 3 months before the blood draw, which is clearly tracks with their decreased immune aging, musculoskeletal, and metabolic aging. And that to me is really exciting. And so again, this you can see that at the 3 levels, we are able to really see how we can customize this at an individual level. So overall, I just want to say that many challenges exist. We need to have insightful biomarkers, effective geroprotective interventions, and responsive aging biomarkers. But we are building sort of system-specific and sort of even sort of metabolite and protein proxies. We are standardizing intervention comparisons, and we are looking at sort of which biomarkers are responsive. But most importantly, we are even trying to translate this in a clinical setting, where we are looking at insightful aging biomarkers, looking at how to build interventional plans using those biomarkers, and eventually see if those biomarkers are responding at an n equal to 1 level. And with that, I'd like to wrap up. Thank you, and I will take questions if there are any.
Sorry.
No, go ahead, Grace.
Well, first, I just wanted to say that this presentation was so informative, and I really was so interested in it because, thinking about my research interest, talking about like computational methods in psychiatric disorders, and thinking about how you guys are kind of going in a direction of like personalized medicine is also so important in thinking about psychiatric use, because in a way you do have individual differences since everyone experiences a general illness differently. So I just wanted to, say thank you for the presentation.
No, I'm glad. And you might find something really interesting. I'm trying to pull this up. We had a review paper recently, which we did talk about sort of geriatric aging and psychiatry specifically. I'm trying to pull up where it is. Anyway, I'll try and email it to you maybe. Yeah, but it might be of interest to you specifically because that one goes into a lot of detail about this kind of work that we have been doing recently in context of sort of psychiatry and aging, and how we could leverage some of the knowledge that we are getting from these aging biomarkers and sort of reapply it to sort of specifically aging. Actually, here, these 2 papers, these would be really helpful for you.
Hi, thank you so much for your presentation. I have question about the pipelines you used for this research, and uh, any software you use or database, is it possible a little introduce which kind of programming, which kind of database or software you use? Because
Yeah.
I'm doing research like this, and I'm here to methylation. I did some chipsec to research and examine the histone modification in DNA. So I want just to know which kind of software or something like this is suitable for
Yeah.
example, in your study for methylation, DNA methylation, which one you used?
Yeah. So we like on a general programming level, we primarily use R. For doing a lot of analysis, we have this in package called Metal Cipher, which I can put in the chat here, maybe. Uh,
Yeah, thank you so much. If
which is a package that we built as part of the Levine lab. We're actually working on updating it as well. Um, uh, some of the more recent developments have not been pushed yet on GitHub. So still working on that. But yes, so that's the underlying package, but there is a lot of sort of above that, there's been a lot of pipeline development, which in some ways is kind of proprietary, I guess. They're trying to set up like a core facility at Yale to help a lot of different people run these services. So I'm a little less aware about that kind of
so
stuff.
if I want, for example, select and know which one is okay for me to analyze my data after do NGS, I want to analyze my data.
Yeah.
So if I want, for example, know how I can know which package or which software is okay for me is good because I saw different package, different software.
Mhm. I guess that's a much deeper question, I would say, Marisa. I would say that probably we should get on a call and chat more about that one, because that would completely depend on your use case and your analysis.
Um,
So feel
so
free to, I'll put my email in the chat here. Feel free to email me and I'll be happy to sort of discuss this.
thank you so much. I appreciate.
Yeah.
Okay, thank you so much. Okay, I'm Marzia, and maybe in I want to do NGS in one of the center. The
Mhm.
first we think about Yale, but Yale University said us mentioned only in the inside students inside we can use NGS. So we want to select another center to do NGS.
Yeah.
And my chipstick products is prepared already. I have them. Only needs NGS, and after that, I need to analyze with uh,
Makes sense.
different softwares or something. Okay, I write your email. Thank you so much. I appreciate. Thank you.
Welcome.
Thank you. Any other questions? If not, okay, I see.
Yeah, just maybe it may be a thought for another discussion, but I just had a question of access.
Mhm.
Like, would we give this or well, we're not going to give this technology to anybody, but w number one, how much would it cost? And so would it be like used more? We still don't have unlimited resources, even in AI. So would we just preferentially give it to younger people to keep them longer, or would
I
the elderly
so
be left out?
yeah, that's a good question. I think this is, yeah, probably above my pay grade, I would say. I think that's a huge policy discussion. Something as a scientist that I think we should be thinking about while developing these technologies. But also between writing grants and doing the actual scientific research, I often don't get the time to. So I think you're raising a very critical question, one that I do not have an answer to yet. I think the first benefit to these interventions and therapeutics and these biomarkers would to be to the elderly folks. So I think my first this thing would be to give them to the elderly folks to allow them to live healthier, longer lifespans. That's my personal opinion, just FYI. But on a more longer, larger scale, I don't know what the correct answer is, to be honest. In some ways, I would also say, right, like, from a biological perspective, an old person's biology is a lot more disrupted, right? So the risk of or the possibility of improving it is a lot easier, right? But for a healthy young person, right, some of these interventions just might not work yet, right? Because what they're doing, and in my opinion, right now, a lot of the interventions that we have are what I'd say are they're just slowing down or aging. So they're slowing the progression of it. Now, if they're slowing the progression of it, the progression is a lot faster if you're older. But if you're younger, that progression is relatively slower, so to speak. You don't age at the same rate when you're 20 versus when you're 50. You age much faster, so to speak. It's not linear. So I would argue that something like a gerotherapeutic that we develop, say, in the next 10 years, would probably be more applicable to people who are older than there are people that are younger. The same is true for something like right now we're seeing GLP-1 antagonists, right? It's being touted as this really exciting drug, semaglutide, which could be potentially a gerotherapeutic as well, just FYI. A lot of people are saying that it could be a gerotherapeutic. And I see the potential of it, but I don't think it's going to get to being a gerotherapeutic for healthy young people anytime soon. It would probably be a gerotherapeutic first. We probably first see it being prescribed. Right now it's being prescribed to obese people, then it will go to probably sort of people with obesity, and then people who are maybe overweight, then people with some smaller conditions, or like the risk of developing obesity or being overweight, and then maybe sort of higher age, like people with higher ages, and then maybe younger people, if the trickle down effect it works. Because the effect of seeing a change in younger people would be a lot lesser. The chance of seeing a significant change. That's just my, again, hypothesis.
Yeah. Well, I'm glad you got your argument ready. It's good.
Yeah.
Thank you.
Awesome. All right. So let's thank Raghav for this exciting research. Thank you for sharing your exciting research with us. And thank you everyone for joining us. I hope you enjoyed the lecture, and I wish you all a good night. Thank you so much.
Thank you for having me.
Thank
Goodbye.
you.
Thank you so much.
Bye.
Thank you. Bye. Have a good night. Thank you. Bye.
Thanks. You too.